if the slides are moving, okay. Are they moving?
Yeah, right now you're on objectives of the lecture. Yes, the slides are moving. Now we're on the next slide.
Thank you. Thank you. So thank you, Kelsey. Thank you, MBCHB. This should be 2, Kelsey, if I'm not wrong.
Yes.
Okay, fine. So I think we can begin our class because there's quite a bit to be done. And this lecture, sometimes it takes a bit of time, but I'll try to work within the limits as much as possible. I have an hour with you, right?
Actually, you have 2 hours.
Oh, it's 2 hours. Okay, I'll try to run this through, within an hour because I also have another commitment after 10. I'll try as much as possible to give you as much as I can. But let's go through hemoglobin structure and functions. So part of the things I think we shall try to, to address today is looking at the structure and the function of hemoglobin as one of the hemoproteins, because there are several types of proteins in the body, but we have proteins that are called hemoproteins, whose role is mainly to support oxygenation of the body. The other one is, I'll also try to mention a little bit about the types of hemoglobins that we have and why they exist. And then, I may or may not mention aspects associated with hemoglobins. I guess that will be a lecture for another day. But having said that, I think of importance for us as a team is to try and understand this molecule called the hemoglobin. And I have started off with this particular structure, or rather diagram, for us to have an idea of where we are headed. Basically, this is the structure that we are going to be discussing. We are trying to figure out how this structure comes into existence and see how it functions. And you'll notice that this structure has a series of things in it. Okay, when we see a red cell, basically the question is, what is in that red cell? And I think part of the contents in the red cell is hemoglobin. And hemoglobin is, again, not just some, small basic structure, but it is a structure that has chains in them. It has iron in it, and it also has something we call the heme group. And the chains are described as polypeptide chains. And there are 2 different types. So you can either get an alpha chain or a beta chain, depending on the hemoglobin that you're talking about. And we shall be discussing different types of hemoglobins. But basically, it is a conglomeration of chains. And these chains are not just appearing routinely as simple structures. They come as, folded structures. And basically, we talk of helical shapes. So basically, this is what we shall be looking into or discussing as we move along. Please, if I'm not audible enough, let me know, because it is good when we are all together. So hemoglobin, basically, from what you have just seen, is an aggregation of 4 globin chains, okay? And those 4 globin chains is what forms what we call a tetramer. There are 4 different chains that come together. So those 4 chains is what forms a tetramer, okay? And basically, hemoglobin is meant to assist in binding of oxygen and the removal of carbon dioxide, okay? And the intention here is really to make sure that hemoglobin is able to function normally and take oxygen from one part of the body to various parts of the body, and then remove carbon dioxide from various parts of the body, for removal at the lungs, okay? And so hemoglobin is an oxygen binding protein, and it's also a globular protein, and we shall be looking into this particular protein, okay? Hemoglobin, as I did mention, basically has 4 polypeptide chains. And these polypeptide chains, sometimes in our descriptions as we move along, will form what we call subunits. And I'll be talking about subunits a little later, okay? And each subunit has 2 different chains, okay? But hemoglobin has 2 chains. They could be alpha chains or beta chains, or alpha chains and non-alpha chains. The beta chains are sometimes referred to as non-alpha chains. So it is important for us also to understand the various terminologies that we might find ourselves in, even as we get into the depth of the lecture. So from a functional perspective then, what is hemoglobin meant to do? As I've mentioned, it is purely for transportation of gaseous elements, either from the lungs or from the tissues to the lung. But more so, hemoglobin has also been known to assist in managing our some metabolic aspects, especially our pH in the body, and we shall be looking at that a little bit even as we go through this lecture. So our mechanisms to maintain our pHs is also a function of hemoglobin. So for the cells to be able to accomplish this particular function, it requires hemoglobin. Okay, for the red cell basically to move oxygen from one end of the body or carbon dioxide from one end of the body to another part of the body requires hemoglobin. Okay, and the most interesting thing, ladies and gentlemen, that we need to be aware of this early is that each red cell has 640 million molecules of hemoglobin. What does that mean? If each hemoglobin has 4 chains, and you have 640 million molecules of hemoglobin, we are as good as saying, you know, one single red cell can carry millions of oxygen molecules. Okay, that is what we are basically saying, that one red cell has capacity to carry millions of oxygen molecules. And so the body uses millions of oxygen molecules in its functioning. There are different types of hemoglobins, as I did mention. Some of these hemoglobins, as I mentioned, may be adult hemoglobin, fetal hemoglobin, embryo hemoglobin. So there are different types of hemoglobins that we shall be discussing. But an adult hemoglobin is described as hemoglobin A, and the combinations in terms of the alpha and beta chains are 2 alpha and 2 beta within the heme groups. And I think I had demonstrated this when I showed you that initial diagram. The molecular weight of hemoglobin is about 68,000 or 64,500 Daltons. And there are different types of hemoglobins, which we shall be discussing a little more extensively. But here I've just demonstrated that there's a fetal hemoglobin we call hemoglobin F. So the adult hemoglobin is called hemoglobin A, and more often than not, we just write it as HbA. Sometimes others write it as HbA1 because we also have HbA2. Okay, so HbA2 is a little different from HbA1. So these are just all types of hemoglobins that you could easily come into during your practice as a doctor. Other hemoglobins may include hemoglobin S, hemoglobin C, hemoglobin. So there are different types of hemoglobins, again, as I did mention, and we will learn more as we look at hemoglobinopathies. Okay. So having said that, then where do we find hemoglobin? And there are different types of hemoglobin. If you want to look for normal hemoglobin in an adult, which I had indicated is called hemoglobin A or A1, the types of polypeptide chains we find in such hemoglobin is 2 alpha and 2 beta. Each of these combinations come together, and when you get an alpha and a beta combined together, it forms what we call a subunit. Okay, so there are 2 subunits in every hemoglobin. Okay, there's subunit 1 and subunit 2, because a subunit is a combination of one hemoglobin chain and another. 1 alpha, 1 beta together combined forms what we call a subunit. Okay, hemoglobin A in an adult is the major type of hemoglobin. That does not mean that you and I do not have other types of hemoglobin, but majority of the hemoglobin in circulation that we all have as adults is called hemoglobin A1. That is what we naturally should find in an individual. Okay, And we know that, this hemoglobin can also be found in children, but majorly in adults, all right? And then hemoglobin A2 is a minor form of hemoglobin that you would find in adults. Actually, the percentage is relatively small, and it is a combination of 2 alpha and 2 delta. Okay, so hemoglobin A2 is 2 alpha and 2 delta. Hemoglobin A1 is 2 alpha and 2 beta. All right, and we shall be looking at the various terminologies that we use. But basically, it's just good for all of us to know that there are various types of hemoglobins, and you'll be learning more about hemoglobins. You'll be seeing pathology associated with hemoglobins and, the various polypeptide chains associated with these hemoglobins. When it comes to fetal hemoglobin is basically 2 alpha and 2 gamma. Okay, so fetal hemoglobin, which is supposed to be found in the fetus, is basically a combination of 2 alpha and 2 gamma. You notice that the 2 alpha is a common hemoglobin, a common polypeptide. All right, the others tend to vary, and that is what gives the variations in hemoglobins. So after birth, generally, the fetal hemoglobin should be replaced with the adult hemoglobin. So our fetal hemoglobin that was assisting us as feti changes to adult hemoglobin as we grow, all right? And this happens gradually as an individual continues to grow. And these are the various proportions of the hemoglobins in an adult. So all of us may be having some hemoglobin A at large quantities, and these are in the ranges of 96 to 98%. Hemoglobin A2, which was 2 alpha, 2 delta, 2 gamma. And here you have 1.5 to 3.2%. And then you have hemoglobin F, all right, where you have between 0.5 to 0.8%. All right. So I'm hoping that as you go through this lecture, we will be learning more about the various types of hemoglobins. Medically speaking, as I did mention, hemoglobin is a hemoprotein. This hemoprotein is found in the cytoplasm of a red cell. So if you are to look for hemoglobin in a cell, you would find that hemoglobin is only found in the red cell, okay? And there are various quantities within which bodies can operate in terms of levels of hemoglobin. Now, ladies and gentlemen, it suffices for me to mention that hemoglobin ranges or values vary from one generation to another generation. It may vary from one geographical area to another geographical area. It may vary from one age group to another age group, one sex to another sex. So one race to another race. Basically, what I'm trying to say is that you'll get various reference ranges attributed to different groups and different settings. So don't stick your mind to a specific figure. Learn the environment you are in. If you're at Kenyatta National Hospital, we will tell you the normal reference range for an adult male hemoglobin ranges from this to that. For females, it's this to that. So try to stick to the reference ranges that have been established within a region. If you're practicing outside the country, the ranges will again vary. So it is good for those of you who are young, who will be practicing in other setups, to understand that hemoglobin ranges may vary from one to the other. What I've just demonstrated in grams per deciliter is what is heavily referenced in a series of literature, but remember, this may apply more in other setups than in our setup. For example, you would find that hemoglobin levels in a female, in a Black lady, may be in the range of 11 to 15, or 11 to 14 point something, or 16. So those ranges may vary one from the other. And it is always good for us to realize that the ranges may vary from one person, from one group to the other. Okay, fine. I'm trying to see whether there are any questions that I can be answering as I move along. So the chat continues. But basically, as I said, each hemoglobin has 4 polypeptide chains, all right? And I think I'll not belabor so much of that. I'll be talking more about that anyway as we move along. So these are the concentrations of hemoglobin, basically, that you'd find in a lot of literature. But as I said, please be aware that these values may vary from one group to another. Okay, it may even vary from one race to another race. Okay, and so as we rightfully mentioned, hemoglobin will carry, or blood generally that has hemoglobin will carry oxygen. But it's also important for me to mention that hemoglobin, if it is in blood, will carry very little oxygen if it is in solution. So basically, the right place for hemoglobin to function, optimally, should happen in the red cell. You don't want hemoglobin in solution to be associated with any excesses in terms of carrying of oxygen. So blood can carry very little blood oxygen in solution, okay? Especially if you have hemoglobin in solution, okay? So you need oxygen to function optimally in the red cell, all right? You can get hemoglobin in circulation, but that type of hemoglobin may not be of much value in terms of oxygen carrying and subsequent release to tissues. So that's why we struggle to make sure we get normal red cells. Okay, and as I did mention, of course, each red cell has almost 640 plus million molecules, and 97% of this hemoglobin gets saturated when it gets to the lungs. So 97% of all the red cells carrying this hemoglobin gets saturated with oxygen, okay, when it gets to the lung. What does that mean? It simply means that when you have red cells going to the lungs, by the time they get to the lungs, they release the carbon dioxide, and 97% of those cells then get saturated with oxygen. So that 97% of oxygen is delivered from the lungs all the way down to the tissues, okay? And under resting conditions, it's about 75% saturation, okay? So generally when you're just relaxed, nothing much happening, up to 75% of your hemoglobin gets saturated with oxygen. And I think this helps all of us then to sustain some level of oxygenation should there be need for flight or fright, for the tissues then to function optimally. Okay, so what makes up hemoglobin? So hemoglobin is basically made up of 2 similar proteins that are stuck together. So you have an alpha protein and a beta protein. These are all polypeptides or polypeptide proteins coming together. So when you get an alpha polypeptide chain and a beta polypeptide chain coming together, as I did mention, it forms a subunit. But you need 2 subunits to form hemoglobin. So when you have these groups of proteins coming together, sticking together, forming, an aggregate together, that is what we call hemoglobin, and we will be seeing how that structure comes into play. Okay, and it is important that these proteins come together, because when we have these 2 polypeptide chains coming together, it enables the hemoglobin to function properly. And I'll be demonstrating a little later the kind of bonds involved between a subunit and another subunit, or one cluster of polypeptide chains against another cluster. Okay, because again, this is what really helps us to understand how hemoglobin functions. All right, so both proteins must be present for the hemoglobin to pick up and release oxygen normally. And as I said, we will be looking at the details a little later. All right, I've already mentioned the various proteins that are of necessity to us when we're looking at hemoglobin, and this is basically the alpha and the beta globin chains. Okay, so the hemoglobin, as I did mention, must be resident within the cell, right? And the cell then has to be surrounded by a membrane which holds the hemoglobin. As I mentioned, hemoglobin in circulation is of very minimal value. So you don't want your hemoglobin to just be circulating in blood, within the solution. You want hemoglobin to be resident within the red cell. So what is this structure of hemoglobin? I'll repeat some things just for the sake of us to internalize certain details. One is I did mention that hemoglobin is a tetramer. And I also explained that a tetra means 4. So you have 4 globin chains, all right? 2 alpha chains, or at least an alpha chain and a beta chain, or an alpha and delta chain, an alpha and gamma chain, an alpha and zeta. So there are different chains that we shall be discussing. But basically, there must be 4 of those chains. If you're looking at the alpha chain, basically, the alpha chain must consist of about 141 amino acids. So if we were to look at the alpha polypeptide chain, it's poly because there are many amino acids. Those amino acids should constitute at least 141 of them if you're looking at the alpha chain. The beta chain, on the other hand, has 146 amino acids. So the 2 chains are composed of amino acids of different numbers, okay? The beta chain being 146. These chains must appear in a certain format, and the format in which the chains appear are described as helical. A helix basically is when you have a twisting of a fiber or a strand. If you have a strand that has twisted, it twists into a shape that is described as helical in nature, okay? And I'll also be trying to describe how different proteins in the body and enzymes enhance this structuring or this change of structure of a chain, okay? So each polypeptide chain is arranged in a helical structure, okay? And there are 8 helical segments. So basically, I'm not sure if I went back, you would be able to appreciate the various segments. So when the helix falls, it falls into different segments. Let me see if I can be able to demonstrate this. I'm sorry that I have to go back. But basically, if you look at these chains, all right, these chains are folded, and each of the folds forms a segment. So you would have a segment, and different segments have been given different letters, all right? So when you have these polypeptide chains folding, they fold into what we call segments. And there are reasons why they are segmented. And the segments have been given letters, all right? Or rather, they have been given names. And the names or designations are A to H. So there are 8 helical segments, all right? As this structure, this helical structure folds. And the folding is important because there are different bonds that create these folds, all right? And the folds then lead to these segments, and the segments are designated A to H. So the reason why this is important is because one of the segments is key in binding of iron, and iron is essential in the binding of oxygen, all right? So the iron part of heme, all right, the iron part of heme is covalently bound to histamine at position F on the H segment, and we will be looking at those positions a little later. So of importance here is that the H segment of the hemoglobin, all right, or this polypeptide chain that has folded, assists in the binding of iron, all right? And this binding is through some histamine, all right? And we shall be looking at that a little later, okay? So how do we synthesize hemoglobin? So basically, hemoglobin and the globin are 2 different components. So hemo basically, is or the heme part is one component, and the globin is the second component. And the globins are based on the various proteins that we've been talking about. The polypeptide chains form the part of the globin chains. But we also have a heme component. And basically, these 2 are important components of hemoglobin, the heme and the globin. So the heme and the globin are produced, in different sites in the cell. Heme, for example, is produced in the mitochondrium. So if you went to a red cell, basically within the red cell or within whichever cell, you have heme being produced in the mitochondrium, and then globin is produced in the ribosome. So if you looked at the cell, and most of you who have done, who have studied cells or have done cytology, will notice very quickly that the mitochondria has its own functions. And then, of course, the globin, component is synthesized within the ribosomes. These processes must be well synchronized. Some happening, of course, within the cytosol, while the others will be happening within the mitochondrium. And so the synchronization allows the heme component and the globin component to come together to form hemoglobin. And I'll be demonstrating again a little later how the two come together. But I think of importance this early is to notice that heme gets synthesized in the mitochondrium, and then the globin within the cytosol or the ribosomes within the cytosol. Okay? And so when you look at the synthetic aspects of heme, or the structural aspect of heme, basically heme is composed of 4 chiral rings. Okay? So there are rings here labeled or designated A, B, C, and D. These are chiral rings. These are rings, all right, that come together and they get bound by nitrogen ions, all right? And in the middle of this ring is ion. And actually, this ion is important through, because it is essential in terms of the binding of the globin. When you have the globin binding to ion at this particular position, because we will also be looking at the position at which ion binds this globin, then you end up with hemoglobin. So this is the structure that is important in the heme component. You have 4 chiral rings that come together with these different areas designated A to D, being bound together by bonds. And within this ring, all right, is ion. And ion, you remember, if you can recall, okay, was bound to segment H in hemoglobin. So it is important for us to remember some of these aspects. But basically, when you get 4 chiral rings and you pull them together, you end up with heme. And I think, again, we shall be seeing how this comes into existence. So when you get heme, which is basically, and heme is nothing other than ferrous protoporphyrin 6. Is it 6 or 9? Protoporphyrin 9. So basically, let me see. Sorry, guys. When you have these chiral rings, the 4 chiral rings, okay, with ion in the middle there, basically it makes these 4 chiral rings be called ferrous, because now there's ion already incorporated in it, and it is a protoporphyrin ring that we are talking about, all right? And so when you have heme, which is basically the 4 chiral rings together with the ferrous component bound together, okay, you end up with heme, which is basically a ferrous protoporphyrin. And this ferrous protoporphyrin is essential for all cells as a prosthetic group in hemoprotein synthesis. Which are the areas where you would get this ferrous protoporphyrin or heme? You will get this kind of combination in hemoglobin. You may get it in myoglobin. You may get it in cytochromes, especially cytochrome P450. You can get it in other enzymes like catalases, etc., and etc. So this ferrous protoporphyrin or heme as a component can be found in various other structures or substances. And the synthesis of heme, occurs 85% of the times in the marrow when you are developing hemoglobin. So ferrous protoporphyrin is basically found and synthesized 85% of the times in the marrow, which by extension means a lot of our red cells having hemoglobin should have this done in the marrow. But 15% can also occur in the liver, especially where cytochromes are found, all right? And the ferrous ion, ladies and gentlemen, that you saw in the 4-pyrrole ring, okay, has 6 electron pairs. So basically what we saw, that ion that was bound in between the 4-pyrrole rings has 6 electron pairs. 4 of those electron pairs are bound to the pyrrole nitrogens in the ring. And I think you were able to see that nicely here, that you have 4 nitrogen, binding onto the ion, okay? And then you also have 1 that is used for binding globin. And this binding of the globin occurs through a histidine residue. So this ion here, 1 other bond, remember there are 6 bonds, 4 have already been bound to nitrogen. But you have another one, another bond that it uses to bind onto globin, all right? And this occurs through a histidine residue. While the other bond is essential in the binding of oxygen. So ladies and gentlemen, this is what I'm trying to say, that the ferrous group within the 4 pyrrole rings, or rather ferrous protoporphyrin, has 6 different electron pairs that are essential in anchoring the ferrous or the ion within the ring. And it also enables the ferrous component to bind onto globin, so that you have hemoglobin or hemoglobin. And then besides that, for the hemoglobin then to function optimally and bind oxygen, the ferrous component or ion requires 1 electron, all right, to bind onto oxygen when it gets to the lung. So what does that mean by extension? What that means is that each of the 4 polypeptide chains in hemoglobin, all right, each of the 4 polypeptide chains that has a ferrous component in it should be able to bind at least 4 molecules of oxygen, because each of those has a ferrous component bound onto it. So there are 4 different ions bound onto each of the polypeptide chains, all right? So if you were to look at the 640 million hemoglobins, you would have to multiply the amount of molecules of oxygen by 4. And that tells you that the body carries large amounts of oxygen using some amount of blood at any one time. Okay? And I think it is important that we try to understand this early. Okay? So what really happens in terms of the synthetic aspects? Okay? Let me see. The ferric state forms. Okay, fine. All right, no problem. I think it is good some of you are checking up on those. I can see people already looking at the Bohr effect. Okay, no problem. I think we will get there finally. So the thing is, there are different stages of synthesis, of the heme component or the globin component, depending on which area you are looking at. But when you're looking at the heme component, basically the heme component and its synthetic capability starts from the mitochondrium. And this occurs when you have glycine and succinyl-CoA in the presence of ALA synthetase being converted to D-aminolavulinic acid, or ALA. And this gets out of the mitochondrium into the cytosol, where you have this ALA being converted into porphobilinogen, hydroxymethylbilane, all the way down to copropophrinogen 3, not copropophrinogen 1, because there are different arms here after hydroxymethylbilane has been formed. And I'll be explaining these steps shortly, where you have the combinations, the various combinations in different parts of the cell, some occurring mitochondrially and others are occurring outside the mitochondrium. So these other stages that are outside the mitochondrium are cytosolic. These are outside the mitochondrium within the cytosol. So when you have ALA, going through various conversions, until you have copropophrinogen 3 that then is taken back into the mitochondrium, all right, for it then to be bound to iron and subsequently leading to heme formation. There are various steps and stages that occur, within the cell. And as I did mention, some of these stages will occur within the mitochondrium, while others might occur within the cytosol. Okay, and we shall be looking at the various stages and steps, as I did mention. And one of the things I'm going to do very shortly is for you to try and remember this. I'll keep coming back to this particular diagram even as we move along, so that then you can understand exactly how the synthesis occurs. Okay, so having said that, then if you look at the erythroid cell, how does heme biosynthesis occur? There are 8 major steps, and again I'll just be trying to go through the diagram that I showed a little earlier. And basically this heme biosynthesis occurs either in an erythroblast, which means this is a cell that still has the nucleus, it still has capacity to utilize whatever else may be necessary from a structural perspective, or reticulocytes that still also have some nuclear remnants, all right? And the synthetic processes can occur both in the mitochondria and also in the cytosol. And there are 8 major steps, as I did mention. Step number 1 is where you are taking deaminolavulинate synthetase catalyzing glycine and succinyl-CoA to form ALA, which I think I did mention. That condensation process, all right, occurs within the mitochondrium. And I think I had already demonstrated this, that when you have glycine together with the succinyl-CoA within the mitochondrium, you end up with ALA. And this is basically facilitated by ALA synthetase, okay? The next step is basically when you have ALA happening or occurring outside, or the catalyzation of ALA within the cytosol through condensation of 2 molecules of ALA to form a monopyrrole. Okay, this is the porphobilinogen ring. And this occurs when you have 2 molecules of water being removed within the cytosol. And ladies and gentlemen, this is basically when you have 2 ALA being condensed together to form porphobilinogen, all right? And this is occurring outside the mitochondrium. So that is step number 2. Step number 3, and of course, unfortunately, just for the sake of those of us who are in the medical world, it is important that we understand that this particular step is essential because ALA or ALAD, this is an enzyme that gets inhibited by lead. So if you can recall, I think last week we had some Kenyans complaining that they need compensation because lead had caused a lot of problems, medical problems to them. And I think this must have been happening somewhere in Mombasa or thereabouts. And because this particular enzyme gets inhibited by lead, so you interfere with heme synthesis. And when you interfere with heme synthesis, automatically you are interrupting oxygenation generally. And oxygenation means you have low hemoglobin, and subsequently the patient becomes anemic. So patients who are lead poisoned basically get affected because lead inhibits this particular enzyme, all right? So the next step is basically where you have, porphobilinogen deaminase catalyzing the deamination and condensation of 4 molecules. So when you take 4 molecules of PPG, it yields a linear tetrapyrrole. All right, this linear tetrapyrrole is linear tetrapyrrole hydroxymethylbilane, okay, HMB. So basically, that is step number 3, when you have this hydroxymethylbilane, being formed as a result of condensation of PPG, okay? And this is important because this particular step can lead to different outcomes. If you have an enzyme at this particular point that is dysfunctional, then you start talking about porphyrias. And I think most of you may be having, lectures a little later on porphyrias. But basically what would happen then is that when you have at step number 4, uroporphyrinogen 3 synthase catalyzing the formation of uroporphyrinogen 3 from HMB, this enzyme is important because it is absent, then causes HMB to form uroporphyrinogen 1 and coproporphyrinogen 1, which is not essential in heme synthesis. So this absence of this enzyme causes HMB to be converted into uroporphyrinogen 1 and subsequently into coproporphyrinogen 1, right? Here, if you don't have that particular enzyme, you are unable to convert HMB into uroporphyrinogen 3. All right. And as I said, this particular enzyme is important in the sense that its absence then causes the porphyrias that we talk about. Okay. And then step number 5 requires that you use uroporphyrinogen decarboxylase or UROD. So the other one was UROS, to catalyze sequential removal of some carboxylic groups and some carboxymethyl side chains to lead to uroporphyrinogen that then subsequently leads to coproporphyrinogen. And this is uroporphyrinogen 3 and coproporphyrinogen 3. In the presence of coproporphyrinogen oxidase, all right, now you have your coproporphyrinogen 3 taken back from the cytosol into the mitochondria. So this coproporphyrinogen oxidase in the mitochondria catalyzes removal of carboxyl groups and 2 hydrogens from the propionic groups of the pyrrole rings to form vinyl groups. And I think basically this is what we are demonstrating. You end up with this protoporphyrinogen ring that is of necessity, because when you chelate this with iron or ferrous ion, then you end up with heme as an end product. Okay. So basically step number 7 is where you have protoporphyrinogen oxidase oxidizing protoporphyrinogen, that then will be bound to ion, and subsequently you end up with ferrous protoporphyrin, where we started from. And I think now you can be able to see how this heme is actually synthesized right from the mitochondrium all the way down to the cytosol and back into the mitochondrium. And as I did mention, any deficiency of any of the enzymes within these synthetic stages will cause porphyrins or porphyrias that then will lead to different outcomes. Okay, so that in a nutshell takes care of the synthesis of heme within the cell. What about the globin? As I did mention, we have various types of globin. Okay, and these various types of globin will bind with heme to form different types of hemoglobin. So basically, for you to form the globin, it's good for us to note very quickly that there are 8 functional globin chains in life. And these 8 functional globin chains are arranged in clusters, all right? The first cluster is what we call the beta cluster, all right? Sometimes we call it the non-alpha cluster or the beta cluster. The beta cluster basically has the beta globin, gamma, delta, and epsilon globin genes, right? And this particular cluster is represented by chromosome number 11. So chromosome number 11, which is the short arm of chromosome number 11, represents this particular cluster of globin chains. While the alpha cluster has alpha, zeta globin genes on the short arm of chromosome number 16. So the alpha cluster is basically found on chromosome number 16. So chromosome number 11 is for the beta cluster, and the alpha cluster is found on chromosome number 16. Okay. And why is this important? This is important because globin synthesis is noted to start quite early in life, and it starts as early as the 3rd week of gestation. Okay, so by the 3rd week of gestation, a baby already starts forming globins. And some of the globins that combine then with heme to form hemoglobin leads to various types of hemoglobins. For example, as an embryo, you can get hemoglobin Goa, and I think you may have learned this in embryology, where you have hemoglobin Goa 1. You can also get hemoglobin Portland. You can get hemoglobin Goa 2, and you can see the various combinations, all right, of the alpha and the beta clusters, all right? And then you also have the fetal hemoglobin, and you can see what the fetal hemoglobin in terms of combinations would have. And then you can also get adult hemoglobin. So globin synthesis is a key component for us to understand hemoglobinopathies and also understand the various types of hemoglobins, as early as the 3rd week of gestation. So having said that, ladies and gentlemen, how do we synthesize globin? So there are 6 structural genes that code for the 6 globin chains, all right? You have the alpha zeta globin genes, which are on chromosome 16, and then of course the others on chromosome number 11. And if you look at the globins, the organizational structure of the globin and the hemoglobins in general, as I did mention, was as a result of 4 globins or 4 polypeptide chains coming together to form a tetramer. So hemoglobin by extension then is tetrameric. Okay, and as I did mention, there are 4 different polypeptide chains, but those polypeptide chains would combine to form polypeptide subunits. Okay, and we've already also mentioned that there are various hemoglobins, that have been combined together, to give us different combinations. And we talked about the alpha globin, or the alpha cluster, and the beta cluster. Okay, the alpha cluster would fall under the alpha-like globins, all right? And there are only 2 of them. And then the beta cluster has, another 4 different types. And these combinations then can lead to a tetramer of whichever type. You can get 2 alphas, 2 betas of each of those clusters, giving you a tetramer. And because of that, we've already mentioned the various types that we can see under Goa 1, where you have zeta epsilon, and then for Goa 2, alpha epsilon, and for Portland, where you have zeta gamma, all right? And then for the fetal hemoglobin, you have alpha epsilon, and for the adult, you have alpha beta for hemoglobin A1 and hemoglobin A2, where you have alpha delta 2. So basically, these are different types of tetramers that you can end up with. And it is good for us to try and remember some of these hemoglobins, especially fetal hemoglobin, because it will be essential when you're looking at some disorders like sickle cell, thalassemia. It is good for you to remember that fetal hemoglobin would have this kind of combination. Also remember the adult hemoglobin, because that might also help us in unraveling some hemoglobinopathies as we move along. So let us then look at hemoglobin, slightly from a slightly different perspective. So we've said hemoproteins are a group of specialized proteins that contain heme in them, and this is, bound tightly by prosthetic groups within. And heme, as we already mentioned, is a complex protoporphyrin component with ferrous ion in it. So it is ferrous protoporphyrin. Okay, we already mentioned that, and we've seen how that has come into development. We also noted that the ion, within the heme group is held in the center, okay, by 4 nitrogen ions. We've already mentioned that. And this particular ferrous ion can form 2 additional bonds, one for binding the globin and the other one for binding oxygen, all right? And I think we've already mentioned that. So it is important that we remember those aspects. And why is this important? This is important because based on these assumptions, hemoglobin, exists in various states. And of importance is that you basically have, globular proteins coming together. And these proteins, lead to different structures. Hemoglobin on its own is a tetramer. Tetra giving us a structure that is described as quaternary, because a quartet is basically 4. And that is what the final product looks like. But before we get to that final product, there are various states in which hemoglobin then, or the various structure states in which hemoglobin can be found. We have what we call the primary structure. And, the primary structure, from what you can see here in this diagram, is basically just amino acids, appearing aggregated together. But you can also, twist that a little bit. And we shall be seeing reasons why the twisting occurs. And basically, when you have the primary structure, then starting to, to twist itself to form this helix that we talked about, then that becomes a secondary structure. So that helical structure, that twisting of the polypeptide or the primary structure, basically is a function of bonds, and that leads to a secondary structure. But when you have this secondary structure twisting a lot more, to form the various segments that we talked about, and then that takes us to a tertiary structure. And when you now combine the tertiary structure, with other tertiary structures to form a tetramer, then you end up with a quaternary structure. So these are the structures that we normally talk about anytime we are looking at the hemoglobin. So the question would be, how do these structures come about? And I think this brings me to a topic on the role of chaperones in protein folding. And chaperones are basically proteins that facilitate the folding of other proteins, okay? So if you have proteins that assist in the folding of a protein, you have a protein that has amino acids. So those proteins that assist or facilitate, a structure that was initially linear to now start changing shape is a function of a group of proteins called chaperones, okay? And there are many types of chaperone proteins. But I think of importance for us to understand is a protein called the heat shock protein. And basically the heat shock protein is a protein that has been tested under high temperatures and found to still allow recreation of a protein chain or a structure. And the reason why I mention this is because when we are doing molecular work in the lab, and you take a protein and you open that protein using heat, you can actually open proteins, stretch them out using heat. You can open them up. But as soon as you leave or remove the heat, the same proteins go back to certain, shapes. And what brings them back is really nothing other than these chaperones, okay? So these chaperones then were described as heat shock proteins, all right? And what do they do? These particular chaperone proteins are thought to stabilize and facilitate the refolding of proteins that were either partially denatured or, were initially exposed to elevated temperatures. So the heat shock proteins or the chaperone proteins literally just stabilize those amino acids, or rather those proteins, and tries to recreate a shape that originally, would make the structure more stable. And there are two different types of heat shock proteins, ascribed as HSP, either 70 or 60, that may be of interest to us, all right? And I think when you are reading, please try to just see how these heat shock proteins assist, in the folding and the unfolding of hemoglobin. But basically, the HSP 70 family or member group of chaperone proteins assists basically the polypeptide or the structure, to remain stable, and you are able to maintain its unfolded states. So when you have amino acids in the primary structure, okay, retaining that particular structure, it is because of the HSP70 family of proteins. But when you want to start twisting it so that then you start ending with a helix, basically this is a function of HSP60 family of proteins, all right? And so HSP70 just maintains the routine amino acid long polypeptide chain, but then you have a combination of these particular HSPs with 70 and 60 coming together, then for you to get a bigger and much more twisted structure that leads to the secondary polypeptide chain, right, which is more of a helix. And so these proteins help us just to try and create different shapes of the amino acids that will have been brought together to form, the polypeptide chains. But this cannot happen without enzymes, ladies and gentlemen. So again, for us to be able to form this nice globin that then will be bound to the ferrous, at whichever position, requires that some enzymes get involved. And I think there are two key enzymes that are important because remember, the chaperones, the HSP 60 and 70, were assisting us to just, recreate the shapes. But then you need enzymes that can assist. And some of the enzymes that have been mentioned include protein disulfide isomerase and peptidyl prolyl isomerase. And these are just enzymes that then assist us, to recreate different bonds within the various structures that we talked about. So just be aware that, the shaping of the amino acids into whichever form or structure is a function of the chaperones and enzymes, all right, that assist us in the folding process. And so the primary structure basically is just nothing other than aggregation of the various amino acids. And some of these amino acids will either give you the alpha chain or the beta chain. And so when you have the two chains, basically, coming in together, and this is heavily supported by HSP70, would give you your initial chains, the primary polypeptide chains, all right, which is, just aggregation of amino acids, all right, to form, the two chains. And you could get any combination. You can get alpha delta, or gamma or whatever it is, for as long as you're just dealing with polypeptide chains, all right? That is the primary structure. But then when that structure, as I did mention, starts to, twist itself through, the aid of HSP60, together with, the various enzymes and the various terminal ends of the amino acids, then you start having a helix. And basically, the secondary structure then gets born. And this is when you have the initial polypeptide chain folding itself, all right? And this happens because there are different interactions that come into play. So the various, backbone atoms, or atoms within the backbone of the structure starts coming together, and you have these bonds, acting in the presence of the various enzymes. Then you end up with a helix. And I think if you can see, this diagram for the secondary protein structure, you'll notice that the helix basically seems to have bonds in between. You have, this carbonyl O and the H, from the amino termini here of this amino acid forming a bond. And so you have this carbonyl O and, and the H coming together basically to create a new structure. And as I did mention, all these are facilitated and supported by enzymes and the HSPs that we talked about, okay? And so you end up with either an alpha helix or a beta helix when the primary structure starts to create this new shape, all right? And I think when you open also this, this particular polypeptide, if you are to open it up, you would also see other, points or regions on the amino acids, either the R, H, or the O regions. We've already said the O and the H, tend to pull each other together to form a helix. But then you also have the R regions And the R regions basically will, use these bonds from these regions, a little later to try and create the tertiary structure, so that then you end up with different segments. So you have a helix that has, refolded to form a segment. And so there are regions that I can demonstrate this early, because if you looked at this polypeptide chain, you have O, H, and R, okay? Areas that would subsequently, assist us with the bonding of the structure. And so when you have the helix on its own, before you start folding it further into the various segments, you have a secondary structure, okay? And so when that secondary structure continues folding and you have a three-dimensional structure that has various segments in it, then that is what we call the tertiary structure. And I think, as I mentioned, ladies and gentlemen, basically this is a function of the HSP proteins and enzymes that assist to shape the various amino acids. Okay, and as I did mention, the R groups, within the various amino acids assist to create, a slightly more, more twisted and folded polypeptide. And I think those R groups then create very different bonds. And there are four major types of bonds that are associated with the R group. One bond is the hydrogen bond, okay? The other bond are called ionic bonds, but you can also get dipolar to dipolar interactions and a whole range of other bonds that can arise from these R group interactions. And I think this is just a demonstration of what the tertiary structure would look like. And as I said, it is a tertiary structure that gives us now the various segments within hemoglobin. And so here you can see different bonds being formed. You have the ionic bonds here, okay? You have hydrophobic bonds or hydrophobic interactions. Of course, we know what hydro means and phobia. You also get, by dipole to dipole, you get disulfide linkages there, all right? And then you can also get other hydrogen bonds in between. And I think the hydrogen bonds, as you may have noted initially, is what gives us the secondary structure. So basically you have an aggregation of different bonds, complicating the structure a little more to form the various hemoglobin segments. Finally, ladies and gentlemen, so what really happens when you have now formed your tertiary structure? You can bring various, groups of these globin chains to form your tetramer. So when you bring these proteins together, so that you combine different polypeptide chains, an alpha and a beta, an alpha and a gamma, an alpha and something else. When you bring those units together, okay, one chain and another chain, either an alpha cluster and a beta cluster, when you bring these two chains together, you form what we call a subunit or a dimer. So dimers and subunits are one and the same thing. Di means two, all right? When you bring an alpha cluster and a beta cluster together, you end up with what we call a subunit or a dimer, okay? Each hemoglobin has 4 chains or 2 dimers or 2 subunits, okay? So when you have 2 subunits or 2 dimers coming together, you end up with a quaternary structure. And basically, the quaternary structure is what we have here, okay? When you have 4 globin chains or 2 subunits, an alpha and a beta coming together forms a subunit. Another alpha and another beta forming the next subunit. So these 2 subunits or 2 dimers is what constitutes a quaternary structure. And so what have we said, ladies and gentlemen, in summary? That the synthesis of hemoglobin starts quite early, where you have ion absorbed, and, moved within the body using transferrin, gets into a cell, and that ion then gets either bound to ferritin and stored, or released and converted into ferrous, or gets into the cell, the mitochondrium, where it will be bound to heme. Okay, and the heme, as we all know, is a function of glycine, all right, and succinyl-CoA, which combines initially in the mitochondrium through some cytosolic or cytoplasm activities, subsequently back into the mitochondrium for it to be bound to ion to form heme. And when this gets out of the mitochondrium, you also have amino acids, going through the ribosomes, leading to formation of the polypeptide chains or the globins, which will be alpha and beta globins. And this then will bind with heme to form hemoglobin. So basically, these are the various steps that we go through when it comes to hemoglobin synthesis, including the various types of hemoglobin. And so this basically makes us realize that hemoglobin is not a very simple structure, but it is basically an aggregation of globins in the presence of the heme group, okay, leading to a normal hemoglobin that then can function to carry oxygen. So this basically is just a demonstration of ion within the hemoglobin. And we've said ion plays a key important role because it has to carry oxygen to and from the tissues so that then we can all function. And we also know that ion binds oxygen to hemoglobin that then travels to the bloodstream, for utility in other parts of the body. Ladies and gentlemen, I think it would be unfortunate if we don't talk about, the role of hemoglobin in, managing our pH in the body. And so we know that hemoglobin also buffers, the hydrogen ions in circulation. And how does it do this? Okay, other than it carrying oxygen and carbon dioxide, it also carries hydrogen ions. Okay, and we know that the binding of oxygen into the hemoglobin is basically described as cooperative. Okay, because hemoglobin binds oxygen weakly, generally at low oxygen pressures. All right, so hemoglobin would tend to bind so slowly to hemoglobin when the oxygen pressures are low. Okay, the binding is pretty slow. And this is good because if you have hemoglobin in the muscle where the oxygen pressures are low, why do you want the little oxygen in the muscle to start binding onto hemoglobin? You want that oxygen to remain in the muscle so that then it can work better there. But you want the hemoglobin to bind onto oxygen in the lungs faster so that it can now do what it is supposed to do. Pick that oxygen from the lung and take it to where it is supposed to be. Okay, so this is a question of pressures, oxygen pressures or partial pressures in different areas, that enables the hemoglobin to function properly. The other thing that is of importance for me to mention this early is that the binding of the first oxygen to any hemoglobin makes it easier for the other subsequent chains or hemoglobins to bind onto oxygen. So the initial binding of oxygen makes it easier for the next step to be bound, or the next hemoglobin to bind onto oxygen. And remember, there are 4 chains, within the hemoglobin that would be requiring oxygen. And so this causes us to, have an effect that is described as allosteric effect, leading to a sigmoid shape where you have partial pressure against saturation of oxygen, giving you a curve, that is described as a sigmoid or an S-shaped curve. And this curve looks at the ability of oxygen to get saturated hemoglobin to get saturated with oxygen depending on the concentration of oxygen. All right. And so that particular curve will help, we will look at it a little later. So I think it's also important for me to mention this early, that because the hemoglobin structure is a tetramer, and it is a tetramer of 2 dimers or 2 subunits. And as I did mention, when you have an alpha beta combined together, you end up with one subunit or 1 dimer. So every hemoglobin quaternary structure has 2 dimers or 2 subunits. And it's also important for me to mention that the bonds between 2 globin chains in a dimer are described as hydrophobic. Hydrophobic means they are water-hating and they are very strong bonds. Okay, and I think we demonstrated some hydrophobic bonds when we were looking at the tertiary structure. So those bonds are essential in bringing together, 2 globin chains to form a dimer. So the bonds between a dimer are described as hydrophobic. When you have 2 dimers coming together, which means subunit 1 or dimer 1 and dimer 2 coming together, they are bound in between by polar bonds. The polar bonds are said to be weak bonds. So the bonds between dimer 1 and dimer 2 are described as weak bonds. They are not very strong bonds. And this is important, ladies and gentlemen, because the strength of the bond determines the ability of the hemoglobin to bind oxygen. When you have the first oxygen through the allosteric, if you can remember, if we said when the first oxygen binds, the other subsequent oxygens easily bind. When you have 1 oxygen binding through the weak bonds, then it is easier for the next hemoglobins to bind onto oxygen. And I think that is what we are trying to demonstrate here. I don't know if we have it. No, we don't have it elsewhere. That basically when you have oxygen binding onto the first hemoglobin, the rest easily binds into the remaining hemoglobins. And so this makes it easier for us to talk about different states in which hemoglobin can exist. It can either exist in a deoxygenated form as deoxyhemoglobin, or gets oxygenated to be oxyhemoglobin. Okay. Now, so hemoglobin then in the body exists in 2 different states. And the different states is basically a function of oxygen. When hemoglobin binds onto oxygen, it changes from a deoxy to an oxy status. The 2 states are either described as taut or T forms or T states, or R forms or R states. The T form, which is said to be tense or taut, okay, occurs when hemoglobin is not bound to oxygen. So when you have a deoxyhemoglobin, we describe that state of hemoglobin to be in the T state. It is in the taut state, okay? And the reason is that the taut state is said to have a much lower affinity for oxygen compared to the R state, which is a relaxed state. So the taut state has very low, affinity for oxygen, and so the tendency is to be more deoxygenated than to be oxygenated. But the moment you start oxygenating that hemoglobin, then you transition it from, yes, someone is raising his hand. Okay, let me see. I'm not sure whether I could be able to help. Punguza mauti sasa. I don't know what that is. Anyway, possibly you guys are just communicating amongst yourselves. And I don't want to be distracted. So the thing is, you have two forms or two states in which hemoglobin exists. And I've tried to explain what the two forms really are. One is a tensed form when hemoglobin tends to have a low affinity, and the other one is relaxed or R form when oxygen molecules start binding onto the hemoglobin. And so you can have those conformational changes, as oxygen gets into the hemoglobin. All right. Having said that, then this is what exactly happens where you have the bipolar bonds getting weakened as oxygen enters the hemoglobin, and that makes it easier for us to transition the hemoglobin from a T state to a relaxed state. Okay, and these are the two forms in which hemoglobin can exist. And I don't need to belabor on this because I think I'd already explained this, that the process of oxygen binding to the hemoglobin is basically, the hemoglobin changing its shape and making it easier through the polar bonds for oxygen to bind. And once the first oxygen binds, it makes it much more easier for the second, the third, and the fourth oxygen molecules to progressively, enter the hemoglobin. So it becomes much more easier, and that gives the hemoglobin much more advantage in terms of its function of carrying oxygen. Okay, and the same can be explained when the hemoglobin gets to the lungs. It's easier when it gets to the lungs where the partial pressure of oxygen is high for hemoglobin then to bind onto oxygen and subsequently deliver that oxygen to the various tissues as it releases carbon dioxide. Okay, so when oxygen then bound oxygen in the hemoglobin gets to the tissues, what happens? As soon as the first oxygen molecule drops off from the hemoglobin, the next is automatically a reversal of what we saw. The next oxygen also drops off quickly, and this allows hemoglobin to release oxygen much more easily and more promptly to tissues. All right, and what this does is then that it allows the hemoglobin to start picking hydrogen ions and we shall be looking at that shortly, and picking carbon dioxide from the tissues and delivering it back to the lungs, and then you have the same process repeating itself as the hemoglobin gets moved in blood from one area of the body to the other. We've already gone through this slide. And so which are the various states in which hemoglobin can exist? We can get oxyhemoglobin, which I've already mentioned, is the R state. Deoxyhemoglobin, when carbon dioxide, when hemoglobin has no oxygen, it is described as deoxyhemoglobin. But you can also get other hemoglobins, methemoglobin, all right? When you are ferric instead of ferrous in the heme group. You can also get carbon monoxide binding onto hemoglobin, and you end up with carbonyl hemoglobin. And this occurs during carbon monoxide poisoning, and this is how carbon monoxide kills. Carbon monoxide, family binds onto hemoglobin, and so the hemoglobin is not available to deliver or pick oxygen at the lung level. So even if you have all your blood circulating to the lungs, the hemoglobin is already bound to carbon monoxide. And so it's family. In fact, you can see carbon monoxide has 200 times higher affinity to ion, ferric ion, than oxygen. So ladies and gentlemen, as doctors, it is good for us to remember that carbon monoxide kills because of its ability to bind onto hemoglobin. Okay, it binds onto hemoglobin, it can't release that hemoglobin, and so it causes death much more faster. But you can also have carbon dioxide binding onto hemoglobin, and this is basically bound easily, and it can be reversed when hemoglobin gets to the lungs. And so you have some amount of carbon dioxide transported through hemoglobin. But you can also get abnormal binding of proteins to hemoglobin. For example, not just proteins, even sugars. You can get sugars like glucose. Glucose can bind onto hemoglobin to form what we call glycohemoglobin or glycated hemoglobin. This is this is abbreviated as HbA1c, and this has been used to measure compliance of medication for diabetics. So we can use the levels of bound hemoglobin to other products to determine the compliance status in terms of management of different states and in different disease entities. Okay, so what are the factors that affect saturation of oxygen? This takes us to the oxygen hemoglobin dissociation curve, which is actually the sigmoid curve, all right? And here we look at the oxygen carrying capacity of hemoglobin at different partial pressures of oxygen. Ladies and gentlemen, as I know this was a very long lecture, sometimes quite boring, and sometimes quite lengthy. I think we shall be finishing shortly. Just bear with me a little bit. So the sigmoid curve, and unfortunately, these are the more difficult areas sometimes to go through. The sigmoid curve, which I said was S-shaped, basically is a demonstration of the binding of oxygen onto the hemoglobin. So when you have one molecule, as I did mention, of oxygen binding onto the hemoglobin, the second, the third, and the fourth is much easier. So initially you'll see a curve that drags and then suddenly rises, because the dragging initially is as the hemoglobin is trying to allow through its polar bonds for the first oxygen molecule to get in. It takes a bit of time. It takes a bit of pressure. But then later, gradually, it rises because the partial pressure of oxygen increases, and you have, the oxygen molecule allowing the second and the third molecules to bind much more easily. And so by at 26.6 millimeters of mercury, which is said to be the P50 or the partial pressure of oxygen, at which at least half of the hemoglobin is saturated, this occurs at a partial pressure of 26.6 millimeters of mercury. And I think this is demonstrated in this diagram, this hemoglobin oxygen dissociation curve, which, as I said, is S-shaped. Okay, you can see an S-shape here, where the Y-axis is the saturation of hemoglobin, okay, in terms of oxygen saturation, and then the X-axis. So the Y-axis is oxygen saturation of hemoglobin, while the X-axis is the partial pressure of oxygen. So the more partial pressure of oxygen you have, it simply means you are dealing with a hemoglobin that is a little more difficult to either let go of oxygen or at least allow oxygen to come into the hemoglobin. And so that gives us certain parameters that we consider. So at 26.6 millimeters of mercury partial pressure, at least 50% of the hemoglobin gets saturated, okay? And so if you come towards the left side of this graph, you would tend to get blood that is less oxygenated, and that's why we talk of venous blood more than on the right side, where you would tend to get more oxygenated blood. And we talk about now venous versus arterial type of blood. But having said that, it's also important for us to note that we have scenarios that makes it easier for oxygen to get into the hemoglobin. Okay, and I think we will be discussing some of those scenarios. And then on the right side of this particular slide is the Bohr effect. And basically the Bohr effect is the low affinity of oxygen to bind onto hemoglobin for various reasons. And a lot of this is explained by, the metabolic issues in the body, where you have the acid-base balance issues within the body. And basically what happens within the body is that in the tissues are high concentrations of carbon dioxide. We know that in the tissues, oxygen has already been used. A lot of carbon dioxide has been built up. And so that carbon dioxide exits tissue and enters the red cell. When it enters the red cell in the presence of carbonic anhydrase, it forms, in the presence of water, carbonic acid. And so the carbonic acid, dissociates into bicarbonate, okay? And it also releases hydrogen ions. It is these hydrogen ions that determines the acidity or alkalinity of our pH. And so the higher the hydrogen ions, the lower the pH, and so the acidity is high, all right? But what I'm trying to demonstrate here really is that when you have carbonic acid being broken down into bicarbonate and hydrogen ions, the hydrogen ions get bound to hemoglobin, okay? So what will happen is that when hemoglobin gets to the tissues, it is the carbon dioxide that then enables this hemoglobin to release oxygen, and that oxygen is taken up by the tissues, while the free hemoglobin binds to the hydrogen ions. And these hydrogen ions then can be mobilized and moved, for removal later. And so the bicarbonate then gets, removed from the cell, and it will go into this chloride with a chloride shift. And basically this effect is what we call the Bohr effect, okay? Where you release hemoglobin, you release oxygen from hemoglobin and bind it with the hydrogen ions. And basically when you have so much of the hydrogen ions, which means you have a lower pH, sorry, what will happen is that you'll need a lot of, oxygen, or there is a higher increase of the partial pressure of oxygen for you to saturate the hemoglobin, okay? And so if you come back to the sigmoid curve over here, you will notice that a lower pH, which means more hydrogen ions, makes it more difficult, and you need more partial pressure of oxygen to saturate, the oxygen. So the same applies to 2,3-DPG, which is diphosphoglycerate. If you have high levels of 2,3-DPG, then you have lower chances of you saturating the oxygen levels. Hemoglobin S is the same. It's very difficult to heavily saturate hemoglobin S. You need higher volumes, higher pressures of oxygen for you to get the same outcome. And the vice versa occurs when it comes to also releasing this oxygen to the tissues, okay? And I think I don't need to belabor on this. Let me just end by saying that the normal position of the curve depends on the concentrations of 2,3-DPG, hydrogen ions in circulation, and that is basically the pH, and then carbon dioxide in the red blood cells, okay? And of course, the other key component is that the type of hemoglobin also plays a key role, and that would be the structure of the hemoglobin in circulation. If it is a fetal hemoglobin, or hemoglobin S and other types of hemoglobins, then you'd have challenges with oxygenation. I think I'll leave my presentation at that, ladies and gentlemen. Kelsey, thank you very much. Let me see if there's anything in the chat box that I need to address. All right. I think most of you have been discussing. I guess the discussions have been fruitful, and most of you then can go read as a group and understand more. Okay. Yes, is the fetal hemoglobin replaced by adult hemoglobin? Again, remember, these are just genes that then helps the transitioning and allows more of the adult hemoglobin to be synthesized based on the various categories of globin chains. I can see someone mentioning the percentages. Okay.
Excuse me, sir.
Yes, please.
Yes, I had a question.
Sorry, I didn't hear any question.
Yes, I'm asking.
Yes, please.
Yes, you talked about hemoglobin. Sorry, about the free hydrogen ions bringing about this. Yes. Now I was wondering, because I read somewhere that hydrogen combines with the hemoglobin to form the hemoglobinic acid, and that reaction kind of acts as a buffer, like helps to maintain the blood pH at a constant. So how does the acidity come about now? Because now I was thinking that the buffer maintains the blood pH at a constant.
Okay, so we can reason together. It's always good when we try to reason together. So remember, the pH is a function of the hydrogen ions, right? Okay, and we all know that hemoglobin assists to buffer this by taking the excess hydrogens. And I think if you look at this diagram here, you have basically carbon dioxide entering the red cell to form hydrogen, to form carbonic acid, right? You can see that. And what do you want? You don't want more acidity. And so you break this up so that you have bicarbonate. Bicarbonate is alkaline or acidic? Hello? Whoever was asking me the question, I don't think I saw your name.
Oh, yes.
Yes, you are who? So that you can have
Brian.
Okay, yes, Fred. So you can see that when you break up this carbonic acid, all right, you end up with bicarbonate and the hydrogen ions. And so bicarbonate is alkaline, right? So you are able to generate these to neutralize any excess, acidity in circulation. And then the rest is taken up by hemoglobin. And that is how hemoglobin helps to buffer unnecessary or any excess hydrogen ions in circulation that would otherwise cause more carbonic acid that would, cause changes in the pH. Okay, and that's how, we demonstrate the role of hemoglobin in assisting with acid-base balance. Okay, and that basically, again, if you look at even the graph, what we have demonstrated is that when you have an acidic environment, you are unable to release the oxygen to the tissues, and that worsens, the metabolic status of the body. Okay, but then when you have, when you have a lower pH, which means you have a higher hydrogen ion in circulation, you are unable to release the oxygen compared to when you have a higher pH, which means less hydrogen ions in circulation that enables hemoglobin to enter, that enables oxygen to enter the hemoglobin much more easily. So if you look at this diagram, and I'll want you to possibly just take a bit of your time and see exactly, and I'm sure you've taken your time anyway to read, and you can discuss with your colleagues, is that when you have this carbonic acid, broken down into the various products, the excesses in terms of the hydrogen ions are taken up. And remember, hydrogen ion is a function of the pH. That is taken up by hemoglobin. And so that hemoglobin then buffers your acid-base balance in a manner that then enables the body to function optimally as more hemoglobin goes back to the lungs for resaturation. So I don't know if that helps, but possibly you can just take your time and see how that happens. Does that help?
Yes, sir.
Okay.
All right, thank you.
Welcome. So class rep, if it is okay with you, I'll share the notes and then you can share with the rest of the team, and you can read a little more and see how these curves get affected by different states and different types of hemoglobins. And I think it is good when you have or take a bit of your time to just understand what really goes on. Okay, class rep, is that good enough?
Yes.
Okay. All right. Welcome, guys. I wish you well and hope to see you soon.
Thank you.
All right, bye.